Hello, good morning, good evening, good afternoon, and thank you for joining this Neo4j Live. Hi, Ben. Welcome to you as well. Thank you for
Hey,
joining.
Alexander.
Hi. So this Neo4j Live is about Crossr, knowledge graph-based drug discovery. It's what it's called. So I'm very happy to have Ben Vosser here with me today, who is the co-founder of Crossr, which is and I guess you will give us in a few moments. You give us some overview of what it is. So I'll summarize briefly in my words, and then you use the better words, I guess. So I think you founded Crossr a few years ago. So it's a small-ish startup out of the UK on drug discovery by using knowledge graph to make the process better, to make it quicker for companies to develop new drugs and new medicine, I guess, for all kinds of diseases, to help the long-winded process, which usually takes lots of time, to make it that just a little bit quicker, to be fast on the market, which obviously helps companies sell more in the short term, but also obviously helps everybody, you and me, if we get well working or the right medicine for the right treatments, for the right applications, and the illnesses faster is obviously a plus for everybody. So I'm looking forward to this, especially because it's a little of a pet peeve project, or not project for me, but interest, I would say. Since COVID, I'm involved in a few things in the life sciences and healthcare industry around with graphs. So I'm particularly, this is a nice project as well to be here. So enough rambling from me. Ben, again, thank you for joining. How's it going?
Yeah, good. Thank you for having me on Neo4j Live. I've seen a few of these before and I've really enjoyed them. Dr. AI. There's one recently on a chemistry library that was also really good. So I'll start by maybe talking a little bit through what Crossr is and kind of the wider landscape of biomedical knowledge graphs.
While Ben gets the screen sharing ready, this is obviously, again, as usual, is an open invitation. So we try to make this interactive and as lively as possible. So if you are watching this live, then please use the chat, chime in, tell us what you think, tell us maybe your experiences. Maybe you're working in healthcare or life sciences and are in the process of research. Maybe you are in the pharma industry and are in the process of actually working on a new medicine for some treatment, and you're interested in this, or you just are an interesting party, so like myself, I guess. Let us know. Let us hear what your thoughts are, and keep it interactive.
Yeah, please do. I'll start with a quote that so at Crossr at the moment, there's two co-founders, me and my co-founder Jamie. My background is mostly computational, so I have a background in software engineering, data science. I worked at some consumer tech companies like Deliveroo. So if you're in a country that's served by Deliveroo and you've ordered takeaway there, then I've helped you out there. And my co-founder Jamie, the CEO of Crossr, has a background in biology and kind of the pharmaceutical industry. And he recently sent me this quote that I think well sums up how knowledge graphs are incredibly relevant in life sciences and computational biology in general. The quote is from Florian Markovets, and it's: computational biology is providing a reference map for biology, an atlas of life that holds together individual insights. And for me and Jamie at Crossr, this was, this quote and also this paper. So I think did a paper. The paper is all biology is computational biology, and that's kind of a thesis that at Crossr we believe to be true. And the idea is that all these little pieces of biology kind of all come together to build up this atlas of life, or an atlas of the cell, that really describes all of life together. And having that wider context of the entire cell and all these individual insights together can kind of help us move forward with medicines and getting therapeutics faster to the patient. So for us, that was a very powerful quote. And you can imagine that if you're looking at a computational approach for building an atlas of life, the best way to model that is through a knowledge graph. And obviously the leading solution for knowledge graphs and kind of graph-based infrastructure is Neo4j. So I'll move on to the next slide. And when I'm talking about knowledge graphs in computational biology, I'm talking about a biomedical knowledge graph. And the primary kind of node types of these knowledge graphs are gene, disease, and drug. So you have a drug that affects a gene. So that will be the drug will upregulate the gene, or it will downregulate the gene, and that gene will have an association to the disease. So there'll be some kind of genetic variation in that gene will have like a relationship to the disease. So by taking a drug and up or down regulating a gene, you're then having hopefully a positive outcome for patients with that disease. And a kind of wider data model for that is on the right. So this is a biomedical knowledge graph by this open source. It's called HeteoNet. It was released in 2017, and it was, in my opinion, like way ahead of their time. So they released a knowledge graph called HeteoNet that you can, I recommend anyone that's interested in biomedical knowledge graphs, this is kind of the one to definitely explore, because it kind of starts with these core nodes of gene, disease, and compound, includes some important like functional annotations. So on the gene, you have which pathways of this gene in molecular function, other Go processes, some other node types around compound, side effect, pharmacological class, how the compound treats or palliates the disease, and symptoms anatomy. And this by itself represents a pretty good base layer of schema for a knowledge graph in the biomedical domain. So I'd absolutely recommend anyone that's interested in biomedical knowledge graphs, this is a great place to start, because it's at the right level of information versus simplicity to do some analytics on knowledge graphs and play around with that. And kind of what this database is built out of is, you can see here that HeteoNet combines information from 29 public databases. So that's genes that are from Entree Gene. There may be some information here from Ensemble as well. Compounds from drug banks. Disease nodes coming from disease ontology. And I'm sure and pathways is another important kind of biological process from Wiki Pathways and Reactome, and the kind of pathway interaction database that kind of feeds into this knowledge graph. And this was released in 2017 and is available they actually have a public Neo4j instance that if anyone wants to try out Neo4j in a public domain, then they can. Here I'm just showing an example, but so if anyone wants to try out a Neo4j graph database in the biomedical domain, then HeteoNet has a publicly available database. You can query Cypher, the domain language for Neo4j. And here I'm going to just run a really quick query for to say, show me all the diseases that have a relationship to the gene ApoE, and you can see here that there are various diseases that are related to ApoE that may be interesting to you. So yeah, if you're interested in just playing around with biomedical knowledge graph in Neo4j, then HeteoNet has a public instance that's definitely a good place to start.
Yeah, that's that's a good point. That's interesting to know for people. I think that's very, I don't know, because you said 2017. Do you think this is, are people still working on this, or is this more like a project at the time?
Yeah, I think this was a project at the time. There are a lot of initiatives that are carrying on at the moment that are more recently updated. Ones I'd mention are Open Targets, which is a gene disease. So if I search for ApoE here, for example, gene disease, a kind of relationship database that allows you to kind of look at the profile of genes. Are they related to diseases? What pathways they exist in, all that kind of stuff, but not in a knowledge graph. So I think one thing we have found at Crossr is that there are a lot of academic initiatives such as HeteoNet that build out knowledge graphs, but without the right the right funding is required to continually update those graphs, which is why you see like pharmaceutical companies or biotechs that have financial incentives, so they're able to kind of continually update those knowledge graphs and keep them up to date. But yeah, as of 2017, I'd say that HeteoNet was a very good example. I think another good example of one that's a bit more up to date is the clinical knowledge graph. This is open source as well. This was I think about a year ago. This is up to date as of about a year ago. A bit of a larger knowledge graph. So compared to HeteoNet, kind of more entities. So where HeteoNet had a schema of kind of gene compound disease and the surrounding edges, the clinical knowledge graph had their schema is a bit wider. So they had stuff like they've split out gene and protein, and they included tissue and possibly cell type and some other entities that made it a bit more of like a wider knowledge graph. Yeah.
I see. Yeah. Okay.
So all these entities build up a biomedical knowledge graph that can help in early stage drug discovery. But at Crossr, we found that there was an opportunity for an enterprise solution for an operating system that assisted with knowledge graph-based drug discovery. By that I mean a platform that has workflows for target identification, target validation, drug repurposing, with specific tasks such as functional gene annotation, identifying which modules genes exist in, and analyzing the results of experiments such as RNA-seq and CRISPR screen hits, which we can go through an example in the platform of kind of how that would work and how Neo4j helps us complete that task. That's good.
Yeah, sounds good.
So this is the Crossr platform.
Yeah. Do you want to? I don't know. Do you need, maybe zoom in a little bit? I think your resolution is.
I'm on the big screen.
Yeah. Yeah, that's better. Yeah, that's cool. Thank you.
So this is the Crossr platform. And what we will look at now is a workflow for, just an example workflow for target identification following a recent paper or a high impact paper that came out in 2018 that provided a systems level framework for drug discovery with a focus on epilepsy. So this paper came out in 2018. It was It was by the Division of Brain Sciences at Imperial and some other colleagues, and it helped to identify a novel target for epilepsy. And as an example, we'll go through how you could use Crossr to speed up this process and perform the same workflow that was done inside the paper, but using our Crossr platform. So leveraging software, leveraging Neo4j to kind of do the same workflow, but faster. So one thing I'd mention is we'll start by importing the data set that was used in Craft. Craft being the analytical framework for target discovery from this paper. And one of the first things that they did note is that they had an interest in focusing on the cell type microglia. There's a couple of reasons why you might want to initially focus on a cell type. One would be commercial reasons. So if you have a pharmaceutical partner, then they may be interested in that cell type. Also, it may be easier to target genes that exist in that cell type with a drug. So we're going to focus on a module of genes from the Craft paper that exist in the cell type microglia. So I will start by importing that module into the Crossr platform. So this is the Crossr canvas. And once I import the modules, this is a module from the Craft data set, which is a set of differentially expressed genes in the disease state of epilepsy that are also enriched for the cell type microglia. And we can add those to the canvas. And in the paper, this was a module of genes that they decided to focus on for identifying drug targets. You can see here, so we've imported the differentially expressed genes into the canvas. You can see here we have a network of genes that has built up an interaction network here. Probably one of the first things I'll do is genes that, as we're trying to look at kind of knowledge graphs and a network of genes, I'll get rid of the genes that don't have a network associated to them, and then start here. So the different colors of genes that you're looking at now represent how differentially expressed those genes were in the disease state. So for example, CD22 here is a very deep red, which means that it was highly upregulated in the disease state, which means it had a much higher expression in samples with epilepsy versus a healthy sample. And ZAP70, for example, here was down-regulated in disease state, so had a much so had a lower expression level in the disease sample versus healthy state in mice, which I think this experimental data set was taken out of mice.
Okay. Yeah. Usually it's mice, I think.
Yeah, and there's a whole thing there. Yeah, one quick sidebar on that is there's been a very so on the animal model versus human model, and a discussion around the benefits of knowledge graphs and computational biology is that there was a recent bill passed in the US that meant the FDA no longer require animal testing for drugs to be approved by the FDA.
Okay.
Yeah. So this is very, very interesting for knowledge graphs because they've added a new section that means that in place of animal models, you're allowed to have non-clinical tests which includes cell-based assays, microphysical systems such as organ-on-chips, which is where you take some part of an organ and then you kind of put it on silicon and you're able to create an environment there, or a computer model, where that computer model could be a knowledge graph that represents the kind of the cell in a disease state. So even on the regulatory front, there is full moving progress towards computational models of disease.
Yeah, I think that's very interesting, because then you can, with the help of these models of a knowledge graph, you can find maybe an application that hasn't been used before for already approved, let's say, medicine. I could imagine, and then you can say, hey, you use this for, let's say, I don't know, fever, but you can also use this for I don't know, headache, and you don't have to do the testing anymore. Is that what it is?
Yeah, I think it's a couple of things. I think first of all, ethically, it's not having to do animal testing ethically is a great benefit, because it doesn't require mice or any other animal to be used in that process.
Yeah.
Also, an efficiency thing. It's a lot easier to build. It's a lot faster to build a computational model using a product like Neo4j of the cell, and in different disease states, if you have the data, rather than looking at animal models. So between those two things of kind of speed and ethical considerations, I think this is a great step forward in kind of accelerating a better version of drug discovery.
Absolutely. Yeah. The less animals are needed, the better it is. That's really good.
Yeah.
A couple of comments here on this. Mark writes, can drive real efforts in drug repurposing. Yeah, I think that's true. Especially there when everything is already approved. I mean, maybe completely new discoveries is probably maybe a little, I don't know. I'm not really a fan of animal testing, but I see their point in some regards. But if it's already something is already approved, and you just need to step out of the originally approved path sideways a little bit, then I think it's totally fine. That saves a lot of time, a lot of animal pain as well. So I think that's only wins here. John says, extends the possibilities of a digital twin work. I think yes, that's probably true. What do you think about digital twin? Is there something?
Yeah. So at Crossr, we think a lot about the concept of a digital twin. We have a forward vision of our platform becoming, a digital twin of the cell. And this act is making digital twins more valuable, in meaning that digital twins where you can interactively, for example, model a cell and the disease state and say, if I up or down regulate this gene, what are the downstream effects? Is it moving towards reversing the disease state? And that evidence is now kind of more widely acceptable because of the Modernization Act in helping pharma or biotech companies move forward with move their drugs forward towards human clinical trials. So moving back to the Crossr platform. So here you're seeing a network of differentially expressed genes in the epilepsy disease state. And differentially expressed genes are interesting because if they've changed the disease state, there's a hope that if you can revert their expression to normal levels, then you're reversing the disease state. There's a lot of conversation around this about are the most differentially expressed genes important, or do you just want to find the causal driver of the disease state that's causing this differential expression and then affect that? but investigating the differentially expressed genes is a good start. And what we're going to do is focus on trying to identify small clusters inside this gene set that have been differentially expressed in epilepsy to potentially identify a target. So the first thing we will do is using the Neo4j Graph Data Science library, which provides some great analytical features. One being Louvain clustering with community detection, and some others around embeddings that potentially we can talk about later if we have time.
Yep.
We can identify we can very quickly identify the modules of genes inside this network. So I'm going to use the cross platform to run clustering. And here, using Neo4j's GDS library, we've identified five clusters of genes, 5 functional modules of genes in our network. And I say functional modules because most of the connections between these genes are interactions, which means that the proteins are functionally interacting and having an effect on each other in a functional environment. So the next thing to do is trying to identify which module you'd find interesting to interrogate. So the paper that we're following, Craft, takes the approach of identifying which modules are important based on the enrichment of certain gene ontology processes and pathways in those modules. And one example function that's important is immune response. So having this before, we can now try and identify which biological processes are prevalent in the modules. So what I've just done there is I've just kind of ran enrichment analysis for all these different types of functional annotation for the modules. And I can look at which ones I think are interesting to investigate. And that will vary depending on your hypothesis and yourself as a scientist.
Yeah.
So I've just expanded it here just so I can see it a little bit bigger. And this was done. We benefit a lot from Neo4j here, having these functional entities such as disease, molecular pathway, biological process, cellular component in our knowledge graph, and being able to use Neo4j to easily query those relationships has allowed us to do this in a fast way that might require a few steps in a Python or R script without this kind of tool.
Yep.
And the module that I want to focus on is this pink one, which is cluster 2. So there I've just ran enrichment analysis for all the nodes separately, but also the individual clusters to try and identify which cluster has some interesting findings.
Okay. Yep.
And it is inside cluster 2, which is this pink highlighted cluster here, that we find a couple interesting processes that as a scientist, I may be interested in investigating further. So this would be, for me looking at this, I'd look at inflammatory response, and inside, I think immune response. Yeah, so inside the original paper, one of the things they focus on is immune response processes per module. So if I identify the immune response, you can see that this cluster has genes that are associated with inflammatory response and the regulation of immune response in red and yellow, respectively.
Yeah.
So as a scientist, that might lead me to believe that that module is interesting for one to investigate as a potential like therapeutic intervention for epilepsy, based on these biological processes and the fact that these genes were already found to be highly enriched for microglia as a cell type.
Okay, yeah.
So if I want to focus down, so I select this cluster so I can get rid of the initial results, and I can focus here on this. Yeah. So yeah, so it's going to focus down on this set of genes here, and I can quickly see if there's any links between any of the targets that I've selected and epilepsy. And you can see there's a few already known links between epilepsy and the genes that I've selected. And because we're looking at epilepsy, another potentially interesting phenotype is seizures. So we can add that to the graph as well. I can see there's one gene. There's one gene inside this module that's also interesting for seizures, which yeah, partly validates the kind of selection of this module to look at further. The relationships, I mentioned, is the relationships between these disease and phenotypes and the targets inside this knowledge graph is based off evidence that comes out of open targets. And that varies in what the evidence is. So just quickly, here's an example of the open targets evidence that's used to associate a gene to a disease. This is for a widely known association between APP and Alzheimer's disease. And you can see here that there's known genetic variations in APP is associated with patients that have Alzheimer's disease, and that will kind of increase the confidence in knowing that there's a relation between a specific disease and a gene, but also there's a lot of other factors they consider, for example, text mining. So if there's recent papers that associate epilepsy with CCLF3 or seizures with ITGAX, then that would be considered in the graph to create these edges. So that's, we've just done that quickly to validate that there's some relationship between this cluster and epilepsy or seizures. So we can remove those diseases now. And now we have a module of genes that are interesting to epilepsy. We can continue following the craft framework to identify a potential drug target for epilepsy. And the way that they move forward in doing that is, after they've identified a module of interest, it is to try and identify upstream regulators that have an impact on the module as a whole as potential drug targets. So you're looking for genes that regulate the genes that are in this module and try and reverse them from the disease state back to the healthy state. And in Crossr, we would do that using the master regulator analysis type. So here we're looking at this module. So if I run master regulator analysis, I've now identified, zoom out a little bit, potential targets that are upstream of the genes that are inside that module. One of which that's been identified is CSF1R, which I can visualize here, which is upstream of these targets, SLC11A1 and ITGB2, as a potential therapeutic target to intervene with that module. This target was the one that was identified by the original paper as a potentially interesting target as an anti-epileptic drug target.
Okay. Yep.
For a couple of reasons. One, because it regulates multiple targets inside the module, and also because it's a receptor. that R at the end means here, which means it sits on the outside of the cell, and it makes an easy drug target to manipulate with therapeutic.
No, interesting. So just so I understand it correctly. So we had this module. We had identified in this module that this is the genes in this, yeah, exactly, in this group of genes here is most likely at least partly responsible for epilepsy and seizures. And now we have looked for potential genes or ways of how can we influence this group of genes in a way to make people healthy again if they are affected. And this one we identified is particularly good because it doesn't target only one, but it targets a couple in this group.
So this module of genes are genes that were, you see they're all kind of red or shading towards red, which means they were all partly upregulated in the disease state, which means they were more expressed in the disease state than in a healthy state, and they have a close interaction network between them, which means they represent a module of genes that are potentially functionally related, because they interact with each other.
Yeah.
So having that information, then we're looking to try and understand how we can reverse the differential expression of these genes to revert back to a healthy state. We look for what genes are upstream of that module to identify which ones are potential drug targets to provide like a therapeutic intervention. And CSF1R is a good candidate because in our database, it's been identified to have an impact on 2 of the genes. Potentially that's more in other databases. It really depends on what data you have, and this is an open source data set we're using for the regulatory network, but if you use kind of commercial data sets, they may have more information on which targets they affect.
Yeah.
And it flows with the paper, which does identify CSF1R out of a selection of upstream regulators, mostly because it is a receptor, which means it sits on the kind of outside of the cell, and it's an easier drug target than other genes.
Yeah. Okay. Yeah, that makes sense. You want to find something that is a good vehicle for the drug, and as easy to get to, and but as an impact on as much as possible. Yeah. The question from Dev Tether, the JAK2 has a blue relationship on itself. Can you explain what that means?
Yes. So that would mean that JAK2 is known to downregulate itself. It's a phenomenon in biology that I'm not as a scientist can explain well, but certain genes have a certain effect on their own expression. So some genes that have a when expressed have an effect on the expression of when that transcribes to a protein. It can reduce or increase the expression of that protein. So it's a self loop of kind of a negative self loop.
I see. Okay, thank you.
Yeah.
There were other questions, but we can let's continue, and 2 questions at the end.
Yeah. So that is the demo that looks to replicate, or at least like explore the findings of that paper. And as you saw, there were other targets in that list that might potentially be interesting, separate to CSF1R. But if you were interested in CSF1R, then you could benefit from a knowledge graph and further exploration. So just removed the wrong gene there. So this was the target that the paper identified, and through upstream regulator analysis, we also identified as a potential target for that module. We're looking at epilepsy. So you can also benefit from knowledge graphs in exploring the kind of functional annotation of that target and the neighborhood. So here I can easily say, if I was interested in this target CSF1R, what pathways does it exist in? In transcriptional regulation by Ventex and other interleukin signaling, which means that might help build your hypothesis of which pathways you're interested in looking at for the kind of disease you're looking at. And you can go further. You can look at which drugs are currently known to impact that gene, and you can see there's already quite a few drugs that are known to have an effect on that gene. I think the one that they tested in a preclinical model here was, I think it was, so this one is PLX 7486. I think they had, find. So this paper did move forward with a preclinical model of epilepsy to validate CSF1R inhibition, and they used PLX 3397 as a compound to do that. I think the way they selected that compound was, it was a compound that they had available to them that they knew had an effect on that target. But you could also use a knowledge graph to explore other compounds that have an effect on that target, and look at if you had a compound of interest, you could also look at what other genes that compound has an effect on. So if what potential side effects there are of using that compound for that target, because drugs are known to be messy and often have an effect on kind of various targets, and not just the one that you're interested in. So you have to consider that in your development cycle. So that completes the kind of example, like target identification of our platform. So like CSF1R was validated in a preclinical model, but would obviously need kind of human trials before it was identified as an actual target of interest.
Yeah, of course.
Yeah.
There was an addition to the Jack2 question. Maybe we can quickly slide it in. Sebastian gave some insights, and he said it's indirect effect mainly through STAT and SOX. So these are downstream from Jack, he says. So I think this is like the how this comes to be. So thank you for that, Sebastian.
Yeah. I also, I mean, that's interesting because I also saw that STAT1 and STAT3 are potential master regulators for these modules as well. So yeah, interesting that that they found that insight as well. So that's kind of the demonstration of the platform. And the other things that, I mean, if there are more questions, I'm happy to answer that. But
there is. Sebastian actually had a question, or at least a comment. He says, I think the FDA likes to see at least dynamical models. That was back, but I didn't want to interrupt. Back from the FDA part we talked about. Ideally, so sorry, I start again. So the FDA likes to see at least dynamical models, ideally parts of ones that have previously been approved. So I guess that makes sense. for your new drug, you don't want to go all in with only your let's say your AI advisor told you, hey, this is great, let's use it, and then it's approved. I guess this does not work. Does the knowledge graph connect to relevant models, for example, biomodels or something else, so to add in some outside information as well?
Yeah, I think it really depends on what data you have available to you, because I think knowledge graphs are incredibly powerful, and I recommend anyone that is interesting you really realize the power of a knowledge graph when you're using them regularly, because you realize how much data is very quickly available to you. Is Alex still there?
Yep, sorry.
Yeah, no worries.
Yep.
So you very quickly realize how much data you have available to you, depending on what you want to do. So what I've experienced is, I mean, you can see there in just looking at this platform, is that there may well be a knowledge graph that's just a kind of interaction between, you may say CSF1R, and you would just want to look at the neighborhood of those targets, and that's a knowledge graph. And you can see here that these gray lines are an interaction with a score between them, and the red and blue lines represent the targets that CF1R up or down regulate. But if you have wider information, adding that to knowledge graph is definitely beneficial. I think there's a lot of benefit in having different knowledge graphs for different disease models. So having a knowledge graph that's focused on Alzheimer's disease, having a knowledge graph that's focused on cardiovascular diseases, cardiometabolic diseases, and having the graphs created there, so that you can kind of have these wider considerations that are very, very quickly accessible, due to the fact that they're sitting in this graph infrastructure such as Neo4j, and they're kind of one query away from getting that data.
I guess that like ties in with some of the challenges you have, right? I mean, you have lots of data available, but on the other hand, you need to incorporate that data, and to build a knowledge graph, you need to see, okay, what there's lots of data available, but you can incorporate lots of data, but then it needs to be on the same level semantically, at least so to make sense to connect reasonably.
Yeah, and I think that's a bigger problem in biology than other domains, because there's a lot of arguments on if, for example, I mean, these two targets, they interact with that they have an interaction in a knowledge graph. So that means the two proteins functionally interact, and they have a confidence score of 0.944, and this is coming from StringDB, which is another kind of great resource for, which I'm sure anyone in the biomedical know the names, but of a protein interaction open source database. But there's a lot of decisions in deciding like which databases you want to consider when building up your knowledge graph. Like our knowledge graph here is built from 8-9 source databases that build up like kind of a wider knowledge graph across all these different biological entities, and even at like the protein interaction level, or the gene regulatory network level, the gene regulatory network, which is kind of a directed relationship between genes. There's lots of different databases you can choose from, and as a scientist, you decide which ones you think are valuable, which confidence thresholds you think you're happy to work with. So if you're happy with a 90% confidence threshold when looking at analytics, then use that. But if you're very strict and you want like a 98% threshold, then you might increase that before you start analyzing that as a knowledge graph.
All right, and then there is one more question from Creek Compendium. Wants to know if you know tools like Drug-like made by Martin Shkreli, sorry, which does apparently computational chemistry. I don't know that, maybe you know it, and ligand docking.
Drug-like?
Drug-like, that's what he writes, yeah. If that is something you are aware of, or is this something, well, if not, then...
I'm not hugely aware of it, but yeah, not aware of it, but there are definitely a lot of open source initiatives. And it looks like one of those that kind of moves forwards with open source initiatives that trying to enrich like the amount of data that we have available to us as as scientists trying to identify novel targets, trying to optimize the best leads for those targets and move forward. So great to see any open source approaches to that.
Yeah, cool. Yeah. And then maybe one question for me is on the future or on the outlook on other use cases other than target identification. You see here for knowledge graphs in this particular field of drug discovery?
Yeah, I think that we are really at I mean that quote that I come back to on computational biology, providing reference map for biology as an atlas of life that holds together individual insights, by Florian Markovets. I think he's a cancer researcher at Cambridge. I think it does. I think we're definitely at a turning point in using kind of these computational approaches to really drive forward drug discovery. Partly from the way we're thinking. I've seen almost all pharmaceutical companies are moving this direction. I know that all the big ones they all adopt Neo4j as a knowledge graph. I think I saw a recent presentation by Neo4j at a conference in Basel I was at that described that well.
Yep.
And so technically we're kind of the technical infrastructure is moving in that direction. The last 20 years we've seen lots of data generated through kind of initiatives such as the Human Genome Project that's really kind of generating that data that's then being able to form into a knowledge graph. We're seeing lots of companies being kind of created around this idea of computational biology and knowledge graph based drug discovery. Some to mention would be Benevolent AI. I think Isomorphic Labs is an interesting one to check out. It's kind of Google's or the DeepMind, if you know those AI guys. They've kind of started their own drug discovery company focused on that kind of computational approach, and I'm sure that at the data infrastructure layer, it's going to be powered by knowledge graphs. So with on the regulatory side, seeing stuff like the FDA Modernization Act is definitely helping all of this coming together to I think really speed up the time from kind of therapeutics and getting that to the patient.
Yeah, absolutely. Another comment from Mark. He says, agreed, maybe use disease-specific knowledge graphs for target ID and larger generic multi-disease knowledge graphs for drug repurposing. So, yeah, I think that makes also sense.
Yeah. I think definitely disease-specific knowledge graphs are really valuable when you're focused down on that. If you can get see, building up these knowledge graphs requires a task of, if you're looking at disease-specific knowledge graph, then you need to have the curation to get it down to that kind of disease model layer. But if you get down there, then I think there's a lot of benefit of being there. And then, for drug repurposing or even target repurposing, which targets might be relevant across diseases, which diseases have similarity to other diseases. So if I'm a company looking at disease area X, can I potentially use all the work I've done so far to focus on another disease, whether that's through the same drug or the same target, then that's where like more general knowledge graphs are useful.
Yeah, I mean, this is now probably thinking a little bit into the future, but I could imagine if you think about a disease-specific knowledge graph, you're almost generating a digital twin of
Exactly.
of the paths of the attack paths of a virus or whatever else, or cancer or something, as well as of the medicine, the drugs that help against it, and then you have the direct way, and you can directly see, okay, where are some weak points, where is it strong, where does it help, how does it work along the full line, and then you have the best scenario possible, but it's becoming very complex, obviously.
Yeah, exactly. You want to you want the perfect situation for each disease is to have an individual model for that disease, a digital twin of someone in that disease state that you can then try and in an in silico model see, like, if I intervene with that target, what's the total impact of that disease state, and as long as you're considering like the whole context, then I think it's important there to think about like what cell type are we looking at, which tissue type are we looking at. If you have all those wider contexts, then you can kind of bring them all together to find and take what is like a lot of individual insights and then build novel insights from like the wider picture.
Yeah, I think that's what's complicated. Like what Sebastian says, the problem with digital twins, to get to the mirroring of biological variability, to get the mirroring of biological variability right, where you again need dynamical models or at least very well tuned statistics. So yeah, I think that's probably super complicated. I mean, the body is complicated.
Yeah, it's messy.
Yeah, it's messy. Yeah, and it does things differently sometimes. So it's not as unfortunately it's not as ones and zeros in this regard. Unfortunately, it's not only ones and zeros like with the digital world.
Yeah, exactly.
All right, cool. I think that was very interesting, Ben. Thank you very much again. Thank you for your questions and comments. If people want to continue to keep the conversation going, how would best people reach you or Crossr? How would they get in touch?
Yes, so I'll just share this slide quickly. So this is how you can get in contact. Either my email or the kind of social media that I'm available at is LinkedIn. There's also a Crossr LinkedIn page. If you want to visit our website, we have a form you can fill out to kind of speak further if you want to get a more in-depth demo of the platform or have a conversation about use cases, that kind of stuff. Then feel free to reach out over all three of these options. And also check out our website. We have we're kind of in the process of posting various blogs. One I did recently on recommendation system to drug discovery that we haven't really spoke about here, but I spoke about at NOS 2022, which was the Neo4j conference. That was really good. So I did a separate talk there on recommendation systems that I think is also interesting if someone wants to check that out. And there's a blog on our website that goes through that, and we'll probably soon publish a blog on kind of this example of epilepsy as a target identification in our platform. So definitely follow LinkedIn and the website, and get in contact via any of those three options.
Cool, thank you. There's another question from Dev Tether. Maybe we squeeze that in. Wants to know if you have thought to expand Crossr after the drug has been discovered to use in trials, to use knowledge graphs and Crossr during the trials, or
it's interesting. So I think knowledge graphs in clinical trials is definitely a great use case as well. I think for us, in clinical trials, I see knowledge graphs most beneficial in stuff like patient stratification. So, trying to identify, which patients are based on their demographic and kind of various other factors would be most relevant for that clinical trial. Maybe there's some stuff with geography there. I think knowledge graphs go even further down the pipeline to end patient care. A great talk recently on Neo4j Live was Dr. AI that looked at a knowledge graph that used that kind of had like symptoms and potential. I think it was a chatbot that was kind of leveraged a knowledge graph to help the end patients. So I recommend checking that out. So yeah, definitely clinical trials down even further there's a lot of benefits to kind of knowledge graphs there that we are thinking about.
Yeah, cool. I posted the link to the Dr. AI session in the chat. I'll add it to the YouTube description also. We had lots of links, so I added them all to the YouTube video description. So if you are watching this, you should find all the links there. So I think that's great. Thank you, Ben, again for your presentation, for the demo in particular. I think that was very interesting, very good to follow and very good to see. It's a cool project, so yeah, check it out. Go to crossr.co.uk, or like Ben said, write him an email. So with that, I think we're at the end of this episode. Again, thank you very much for watching. Thank you very much, Ben. I'll be back with the final episode of the GraphQL Book Club tomorrow, Wednesday, at a little earlier than the slot, an hour earlier, 4 p.m. Central European time tomorrow. And on Thursday, another Neo4j live stream. This is a very busy week on a book release, graph data processing with Cypher, with my colleague Ravi, who wrote a book on the topic, and we'll do an overview of this. So that's happening on Thursday. Yeah, with that, I wish you all a great rest of your day. Have a good evening, or enjoy your working day, and have a good rest of your week. And see you soon at another of these sessions. Like and subscribe as always, and see you soon. Thank you, Ben.
Thanks, Alex. Thanks, guys.
Thank you.
Bye.
Bye.